Okay, hi everyone, and welcome to another Win Neuro seminar. And thanks for joining us on this hot Friday afternoon. It's very kind of you. Today, we're really happy to have Alice Berners-Lee with us. Alice is a PhD student at David Foster's lab, previously at Johns Hopkins, currently in UC Berkeley. And she's going to talk to us about some really cool hippocampal replay work. Just a really quick reminder, by now you probably all know this, but about the ways you can ask questions. So on your right hand, there's a chat where you can ask small clarification questions that maybe somebody from the audience can help you. And Alice will also, I'll refer burning clarification questions to Alice. If you have deeper or more profound questions, you can use the Ask a Question button at the bottom of the screen. And we'll go through these questions at the end of the talk, and I'll invite you to the screen. If you don't want to be invited to the screen to ask your question, please say that in the question. Please just have a little note. Great. So with that, Alice, thanks again for coming, and the floor is yours.
Yeah, great. Happy to be here. Hi, everyone. All right, I'm gonna
share your screen.
share my screen.
Yeah.
All right.
Oh.
There we go. Is that better?
Yeah, that's good.
Great. Okay, so yeah, I'm Alice. I'm gonna tell you about how replay slows down with experience. So first, I'm just gonna do a bit of background on replay and the motivation for asking the questions that I wanted to ask. And then I'm going to tell you how replay appears after a single experience, how it slows down with subsequent experience by adding detail to the representation. And then I'll touch on a potential mechanism for this and a potential consequence. And I'll just talk about just the implications that I think this work might have. So yeah, put any clarification questions in the chat, especially early. I don't want to lose people when talking about any of the early analyses. So just as a refresher about replay, so our lab and many others study play cells in the hippocampus. So here's 4 on the right side of the screen in a 2D environment. These are 4 separate cells. And then in a more recent example, over 100 cells recorded simultaneously. So each place cell has a field on this linear track. And so what that means is that if you record from enough of them simultaneously, then you can tell at any moment in time where the animal is. And so what we do often is just simple Bayesian decoding, memory list Bayesian decoding, nothing fancy, no prior, anything. Just looking at each time bin and saying, given the place fields of the cells that are firing, what is the representation of position in the hippocampus at that one time? And so you can ask that for any time bin, and you can move along the entire behavioral session that you have and ask what the representation of position in the rat's hippocampus is at any one moment. And so when you do that, when the animal is running, here's just an example. On the y-axis here is a position along a linear track, and here are a whole bunch of cells that were recorded simultaneously. As the animal's running across the track, you see the cells, they're ordered in the order in which their place fields are along the linear track. And so you can see that they tile the space really well. And this really simple Bayesian decoder picks up where the animal is in space. So here is the posterior probability of where the animal is every 200 milliseconds. Then when the animal gets to the end of the track, there's a little reward there. And so they're eating their reward, and you see these huge, these huge bursts of activity of a whole bunch of cells firing at once. And this is where replay occurs. So this happens along with the sharp wave ripple at around 150 to 250 hertz. And so if you look more carefully, instead of decoding at a 200 millisecond time window, if you look at 20 millisecond time windows, shifted by 5 milliseconds, and do the same Bayesian decoding, you can look at what the representation of this huge burst of spikes is, and see that it's actually a structured sequence of neurons here from another session, just showing that they are representing the trajectory that the animal has just experienced. And so here you can see this representation across time of this probability of position just sweeping across the track really nicely. So one thing that we think that replay might be good for is memory, hippocampally dependent memory. This is just 2 examples of a forward replay and a reverse replay. So the replay after an animal experiences a track can go the same direction as the animal, or go in the reverse direction the animal just ran. And just this, this reactivation of the experience that the animal just had makes it a good candidate for memory. And one of the hippocampally dependent memories that we think that replay might be involved in is episodic memory. And having this kind of mental exploration of a space makes replay a good candidate for episodic memory that takes place in space and time, and is kind of a traversal of an experience. So with that in mind, the motivation for looking at this data was to see how flexible replay could be. Episodic memories, are memories that happen of things that happen once, and then you have long-term memories for these episodes. And so, is replay flexible enough to be able to represent an experience that an animal only had once? And then, when the animal has more experience in a new environment, how does the representation, how does the replay change with experience? I guess if there are any clarification questions now, let me know.
I think you're good. Yeah.
Okay. Okay. So the first experiment I'm going to show you is data that was collected by a former postdoc in the lab, Brad Pfeiffer. So he let animals. So we have a pre-rest where animals are resting in a sleep box. And then he let the rats, explore a novel track for only 1 pass or 2 passes on this novel track. So either half a lap or a full lap. And then took the rat off, and allowed the rat to sleep in the sleep box. And then afterwards put the rat back on the same novel track, and then allowed a full run of the animal to continue running back and forth along this linear track. And so here are some example candidate events. So what we do is look at periods where there's a lot of spiking, and we call that a spike candidate event, spike density candidate event, and then we decode those candidate events. And so you can see here this nice replay that depicts a trajectory across space through time. And we pull out these nice replays by looking at both the weighted correlation of this posterior, so it's like how tight the posterior is here, as well as the maximum jump distance in between bins that the replay takes. So here, and this is a proportion of the track. So here, this is probably the biggest jump that this replay took, and it's about 1/3 of the track. So here are some examples of candidate events that are not replays. So this has a very low weighted correlation, even though it has a small jump distance. It's moving around slowly, but it's moving on all over the place. Whereas this has a strong representation, high weighted correlation, but is this huge jump across the track. So it's really just a representation of one side of the track and then a representation of the other side of the track, and you don't have this smooth trajectory. And so here are just some example replays from one session. These are the 3 best replays from the pre-rest before the animal ran on the track. And you don't see replay, so you see events that have lower weighted correlations and max jump distances. But after just 1 pass across the track, you see these structured replay events that have high weighted correlation and low jump distances. So we quantify that by comparing the number of events that we see that pass these criterion to a time bin shuffle, where we shuffle the time bins here of the decoded posterior, and then see how many replays pass each criterion. So here on the X and Y axes of these significance matrices are the two different criterion that we use. So good replays are in this green box. They have high weighted correlation and low max jump distance. And here the red values indicate that there's a significant number of replays than you would expect by chance by this time bin shuffle. So whereas you don't see replay before the animal has been on a track that it's never experienced before, with just a single pass across the track, you get a robust amount of replay. So this is really exciting because it means that replay is flexible enough to encode a single event. And this rest period is like in a different location. So this is remote replay of this environment, and it goes on for an hour. Another way that we looked at how quickly replay appears is if you don't take the animal off the track. So this is across the first 3 passes, first 4 stopping periods when the animal is running on a novel track. So here is when the animal is placed on one end of the track before it's run across the track, and you don't see any replay. But then as soon as the animal's had any experience, you start to get a significant amount of replay within this green box. So whether the animal is local or remote, you get replays of an experience by just a single pass across the track. So then what happens with subsequent passes? So this is an example of the behavior. So time on the x-axis, position on the y-axis, animals going up and down this novel linear track. And then I've highlighted 7 replays that are significant replays pulled out by those criterion of high weight correlation, low max jump distance. And what you can see just by eye at these events, so these are the decoded posteriors of the events, and underneath I've just put the spiking of the neurons with a few cells highlighted in different colors, and the cells are ordered by where their place field is on the track. So you can see that early on in experience, these replays are shorter and they are rapid. They have high slope, right? They just jut across the track really quickly. And with subsequent laps in this novel environment, these replays slow down. So they take longer to traverse the same distance. And this is across all of these sessions. This has over 1,000 replays in it and 39 different novel sessions. And you can see that the duration of replay goes up across passes on a novel track, and the slope of the replay goes down. So across laps, these replays are slowing down. So we looked to see whether this was something that happened across any experience or whether this was unique to a novel environment. And I looked,
sorry, there's a question about 2 slides ago, I think. I'll read this question. It's from Eitan Shechtman. It seems as though the increase is not specific to replay, but more general. There is an increase outside the green box. How do you explain this general increase?
Was it on this slide probably, maybe? Yeah, so these are 2 gradients of cutoffs. There's no perfect cutoff what you're looking for. So these events are going to be going from really crappy noise and these stationary events towards something that is closer to replay. And so when you get really nice replay here, you tend to also have a significant amount of replay that passes all of these criteria. So if a replay passed all of these criteria, it also contributed to passing this criteria and this criteria. So you will get, a significant amount of all of the types of replay. But this is really the only thing here that is replay. Does that clarify? No.
I think so. Yeah. If people are still confused, then please let us know in the chat. Yeah. I think that was clear. Yeah.
Cool. Yeah, and this I'll just also note that this technique of looking at replay was used by Ting Feng, and there's a paper from our lab that goes through it, and she did a whole bunch of things like looking at what Poisson firing looks like in this and stuff. So if you look at that, it's kind of easier to kind of wrap your mind around like what happens with different scenarios with this kind of analysis. Okay, yeah, so I looked at other sessions where the track was not novel. This includes sessions where this is the second time the animal has been on the track. They've been on it for one run, one full session, and then another day they go on it again. So these are still can be like relatively novel, but it's not the first time that the animal's been on the track. And you don't see that replay slows down in familiar environments as the animal's running. So then one thing I wanted to look at is, whether this is the brain state of novelty that is making these replays slow down. You could imagine that maybe there's something about the state of the entire brain that the time constants are somewhat different, such that every burst of activity is just going to be shorter during novelty. So I looked at those candidate events that didn't pass criterion. And so here in gray are spike density events that didn't pass criterion. So here it's 0.6 for weighted correlation and 0.4 proportion of the track jump distance. And for all of the analyses, that's the cutoff I'm using. And you can see that while the significant replays slow down and those events increase in duration, these spike density events that didn't have replays in them don't slow down, don't increase in duration, don't start off shorter and then get longer. And so we think that this is really specific to this, to the replays on the track. And one thing that can often happen with these events that are spike density events that don't get decoded as nice replays is that, it's the hippocampus can replay remote environments. So it could be that we don't have the right map for these replays. And that's why we're not pulling out any structure within them. So then I looked at some data where animals were allowed to run on a novel track in session 1 and then allowed to rest in the sleep box and then allowed to run on a different novel track in session 2. And I wanted to see whether the slowing down effect that we saw during novelty was specific to the replays of the environment that the animal was actually accruing information in, or whether it was that any replays that are true replays are being changed by this novel experience. And so this here on the left is replays of track A while the animal is on track A. And so you can see, again, that these replays slow down, they increase in duration. Then in track B, these are replays of track B while the animal's on track B, and again, they slow down, increase in duration. However, if you look at replays that are of track A while the animal is on track B, so remote replays of track A in session 2, these replays don't slow down, they don't increase in duration, they don't decrease in slope. And both of these interactions are significant in that the replays of track B while the animal's on track B slow down significantly more than any replays of track A while the animal's on track B. And also replays of track A slow down more when the animal is actually accruing information on track A than when the animal is in a novel environment but isn't actually actively getting any new experience in track A. So it seems like the slowing down effect is not just a brain-wide effect or just a general novelty effect, but seems to be related to the actual experience that the animal is getting by running these laps. One thing I wanted to look at is whether this was true, whether replays started slowing down with any kind of novelty. You might imagine that, if the novelty of a new environment, you need these rapid replays in order to, update a value function really quickly or just explore your mental map because something has changed in the environment, that maybe that any change in the environment should trigger these type of rapid replays. So I looked at data that was recorded from a former graduate student in the lab, Ellen Ambrose, in an experiment where one side of the track had an unexpected increase in reward. The one side of a linear track had 4 times the reward than the other, and it was unexpected to the rat. And did not see that replays changed their slope or duration with this unexpected reward. So it looks like it's just environmental novelty that is triggering this slowing of that's triggering these rapid replays in the beginning and then the slowing down with experience. So another hypothesis that you could have about how this might be working is that maybe having a novel experience, having just one pass across the track that we know can give you replay, maybe that sets off a chain of events within the hippocampus that slows replay down. And maybe it's not this lap by lap experience that you need to slow replay down, but it's just time from having this one experience. That the hippocampus can do it offline, can slow these replays down and with just time. So I looked at back at this data that Brad collected and looked at this rest period in between when the animal just ran one lap on the track and when they got to run a full session on this track. And what I found is that the duration and the slope of replay remained constant across this rest period. Then when the animal was placed on this novel track again and allowed to run more laps, then these replays started to slow down again. So even though the animal had had a single experience and had replay within this rest period, this effect of slowing down was not happening until the animal was allowed to get more experience in the environment. And you could imagine that, well, maybe this just isn't enough experience, right? Maybe just one pass across the track isn't enough to change the weights enough to make this mechanism happen. So I also looked after an animal has had a full session in a novel environment where the replays slowed down, and then looked in subsequent rest and did not see any slowing down of replay, either the duration or the slope. So it looks like time alone after experience, either a little amount of experience or a lot of experience, does not allow for the slowing of replay. And instead, it looks like it's this, it's actually the experience that the animal gets running across the track. So here's another point where I can take any clarification questions before we go into the details of replay. I'm kind of going to zoom in and talk about how the replays are slowing down at a smaller scale.
Yeah, I think we're good. I think it was very clear. Yeah.
Cool. Okay, cool. Yeah, so I've shown you that replay appears after a single experience and then slows down, and that this is specific to local replays in a novel environment, and it's specific to environmental novelty, and that it seems to be experience seems to be necessary to have these replays slow down. So replays don't move smoothly across an environment. This is work from the former postdoc in the lab, Brad Pfeiffer, looking at replays in both an open field and on a linear track. And what you can see here in both, this is just the whole replay collapsed in time, and this is the movement across time. And you can see that there are these jumps in the representation. So the replay hovers and then jumps, hovers and then jumps. And it does this to the slow gamma rhythm, which is about 25 to 50 hertz. So both the spike probability and the step size is locked to slowed gamma, both in open field and in the linear track. And so this is thought to be a signature of an attractor network that is underlying this mechanism. So you could have maybe attractors within this map where the replay is hovering, dwelling, and then moving on to the next position within the replay. So I wanted to see whether this was true in this novel linear track data as well. And this is just an example, and this is across all of the sessions that I see the same thing where replays hover and then jump and tend to hover and then jump to this slow gamma rhythm. So then the question becomes, well, if replays are moving in these steps, these hover jump steps to this slow gamma rhythm, how are these steps within replays changing while the replays are slowing down? So you could imagine maybe if this is sort of a schematic of an original replay where you have this hover and jump within this gamma cycle as it moves across the track. You could maybe imagine that you might get a change in the duration of your jump, where you get a longer jump, or maybe the change in the duration of your hover, where you have maybe strengthening attractors so that you get pulled into this hover for longer with experience as these replays get longer as they slow down. Or you could imagine that maybe the steps themselves are getting wider, so maybe the gamma frequency changes, or these hover and jump steps take longer to get across the track. We found that none of these were the case. And instead, what seems to be happening is that the temporal dynamics of these hover jump steps, these gamma steps, remain the same. The replays just add more steps. And so here's a schematic of what we think is going on. So whereas here there's hovers and jumps of 3 different steps, in a later replay that takes longer to move across the track, you have more steps, but each step doesn't go as far across the track. So the replays are slowing down by adding more steps, and those steps are smaller. So the number of steps goes up and the step size within the replays go down. So this introduces kind of a confusing problem, because it means that if we think that these hover places might be related to attractors in a network, then how are we adding hover locations, right? How are we adding these attractors? Are they moving around, or are they being inserted into replays? So I wanted to get at that. And so first, I wanted to see whether replays tended to hover in similar locations to other replays across within a session. So this is so I took all the replays. This is from an example session, and I just take a histogram of the positions in which the replays hovered. And so in this example, you can see that there are places here I've highlighted with these little dotted lines, or these places along the track that tend to have, that replays tend to dwell in. And I've done a bunch of different control analyses that I'm not going to talk about unless people want to control for the fact, to any questions that I had about whether this could be an artifact of, the Bayesian decoding, or the place fields that we happen to be recording from that day, or something like that. So I can talk more about that if people are interested at the end. But for now, I'll just say that that this bumpy dwell distribution, this hover distribution, isn't dependent on the fields or anything. It seems to be, that the replays tend to hover in these locations. So then next we can look at, okay, this is all the replays for the whole session, but we know that in this novel session that early replays are hovering less, are because they take less steps across the track than later replays. So do they tend to hover in the same places, or are those hover points, those attractors, moving around? And so this is from a single session, and I just split the replays from those first 14 passes up across equal quartiles. And so these are the earliest replays, and these are the latest replays. And when you look at a histogram of where those replays tend to dwell, they seem to be pretty consistent across all of these different speeds of replay. And so across the whole population, I looked to see whether they were more consistent than you'd expect by chance by just taking the correlation across these 4 quartiles for each session compared to circularly rotating these distributions, these hover distributions, so that you say, with the same statistics of how bumpy these are these hover distributions more correlated than you would expect by chance? And across all of the sessions, you see that they're far more correlated than you would expect by the statistics of how bumpy they are. So this seems to point to the fact that these hover locations are pretty constant, even though the replays are slowing down. So this, this means that maybe one of two things could be happening here in the first few laps in a novel environment. So on the left, this insertion hypothesis, the idea that maybe these attractors are being inserted in an experience-dependent manner. So here on the first pass, you could imagine that these are 3 replay events that you have, and these are just some hover points within the replay. So this replay hits this place, then this place, and this one, and this one. So they each only really hit a few places along the track. And if you look at the histogram of where they tend to dwell, there's a missing point. There's a missing hover location. Then they have experience, they go another pass along the track, and then all of a sudden the replay is able to fill in this hover location. And then in subsequent laps, you see replays that are able to hit all of these hover locations in one replay and really slow down this replay. The alternative hypothesis would be that early on in the first lap, you have replays that each individually only hit a few places, but all of the hover points have already been represented. They just haven't been stitched together into a slower replay yet. So if you look at a dwell distribution of these first lap replays, you would still see all of the hover points represented. You just didn't have replays that had all of these, in this example, 5 hover points represented. Then with more experience, somehow the hippocampus is able to stitch together these replays into slower replays that hit all of these 5 hover points. So this is testing whether these hovers are being inserted in an experience-dependent manner, or whether there's just some stochasticity that happens, and then the network is able to stitch these hover points together. And so this is an example session. This is the first pass, second, third, fourth pass, and these are exactly what the schematic showed before, so a distribution of where the replays hover. And what you can see by eye is that in this first pass, there's really only 4 places that these replays hover. And so across all the sessions, I look to see whether there are fewer places that the replays tend to hover than you would expect by chance. And here I'm shuffling all of the replays that would happen later, pulling from the distribution of replays that would happen later. But because there are fewer replays on the first lap and there are fewer hover points within those replays, I also keep consistent the number of hover points and the number of replays. So keeping the number of replays and number of hover points, do the replays that happen on the first lap build a distribution of hover locations that have fewer, attractors than you would expect by chance. And across all of the sessions, I find that there are fewer hover points than you would expect by this shuffle distribution. So what it looks like is that insertion hypothesis might be happening where on the first pass, the hippocampus is not able to hover in certain locations. And it needs more experience, another pass across the track in order to fill in those locations that it can hover in, that it can dwell in replays. And then slowly it will put together the replays so that it hits all of these hover locations along the track. Okay, so another point for any clarification questions. That was kind of a lot.
I can ask. So I wonder. So what's special about these hover points? Why have you managed to find anything that why they're the ones that are chosen to hover?
Yeah, that's a good question. I haven't. I don't know. You could imagine that it could be something that is tied to behavior in some way. Like maybe there was a newly potentiated field from a head scanning event or something that happened on the track. I haven't seen any evidence that there's any correlation with behavior at all, but I haven't done a deep dive into intricacies of the behavior. But it also could be something that is just kind of random within the network, or about just when certain cells come online, certain place fields come online compared to other ones. It might not be actually that tied to anything in the environment. But yeah, I don't know. It's a great question.
Okay, cool. And there's a couple of other questions. Yunze is asking if you think that this is specific to change of replay, or could it be changes to the place field?
Yeah, so I was worried about that. So I have done the analysis 2 different ways. And the way that I'm showing you now, all of these analyses are that I build the place fields lap by lap. Because we know that place fields change across in a novel environment. They shift around and their sizes can change a bit. So I use the map constructed from fields that were only in the last 1 to 2 passes. On the first stopping period, it's just 1 pass, but every other stopping period, it's the last 2 passes, so the last lap, to build the encoding model for build the place fields to then do the decoding. So I don't think that this is because of fields moving around on the track, but it is true that both of those things are happening, right? Both of these things are part of a changing network that's happening during novelty.
Cool, thanks. And one more question. Do the replays get longer in duration with experience?
Yes.
Yeah.
Yeah, they do. So their duration increases and their slope decreases. So they take longer to travel the same distance. Yeah.
Cool, I think we're
cool.
good.
All right, and so now I'm just going to touch on a potential mechanism, one out of many possible ones, one of the only ones that I could test with this data, and then a potential consequence of this effect. So one idea I had is that maybe inhibition is allowing for rapid replay, so lowered inhibition during early laps could be allowing for rapid replays, and then increased inhibition in later laps might be allowing for these longer replays to dominate. So the idea would be that there are representations of positions along the track in early on in experience. Place cells that fire near each other, they haven't had as many theta sequences, they haven't had as many replays, there hasn't been as much behavioral timescale plasticity, and so their connections between these places within the map aren't as strong. However, there's low inhibition, and so the representation is able to jump in between these connections that are weaker. Whereas later there's higher inhibition, so you're not able to have these larger jumps, but you've had all of these sequences, and so all of this plasticity that has really strengthened the connections between these locations on the track. And so you're able to move across the track because your connections between these locations can overcome the higher inhibition. Is the idea. So I looked at inhibitory neurons that we recorded from simultaneously, and here's just an example neuron, and here it's broken down into the first 8 laps and then the rest of the laps. And what I saw is that that inhibitory neurons tended to fire less in early laps than they did in later laps. And so if I looked at the correlation between how modulated they were to replays, this is from the start of replay, the correlation between how modulated they were and the lap that modulation of that replay took place, I saw that overall across the population of interneurons, there's this positive modulation where you had less modulation in early laps and more modulation in later laps. As you can see by the y-axis, we didn't have a lot of simultaneously recording interneurons. There are huge amounts of pyramidal neurons in these data sets, really beautiful data sets recorded from prior people in the lab, but for some reason not a lot of interneurons. So I took advantage of a data set that was publicly available from the Bujati lab and did the same analysis in their data. This is data that was recorded in novel linear environments, and they have way more interneurons and replicated the same analysis here. So it looks like maybe there could be lower inhibition early in learning, and maybe this is involved in allowing for rapid replay. And then this raised inhibition could maybe help this transition from rapid replay into slowing replay. So then when I was thinking about why this would be good for a system, it felt pretty easy to come up with a reason why you would want to have rapid replay in a novel environment. Like I mentioned before, if things are new, you don't know what threats are, you don't know, you just experience one reward, and now you really need to update how you got there so that you can go back to get that reward next time. It makes sense to want to have these rapid replays that quickly explore this map of space. But then why would you want to slow them down and add more detail later on? And so one idea, it could be that, replay is affecting the whole rest of the brain, and areas that are either doing computations on top of replay or are engaged by replay it might be beneficial for those areas to have more computation time. And so the prefrontal cortex is an area that is modulated by replay. And so I took advantage of some data that was recorded by a former graduate student in the lab, Jiajing Wu, with simultaneous hippocampal and prefrontal cells, and looked at whether prefrontal neurons are modulated longer to longer replays. It's pretty simple, and this is just showing a median split on the replays, and this is just showing a z-scored average of all of the prefrontal cells, and you can see that these longer replays have this prolonged activation of prefrontal neurons. So however way that these prefrontal cells are interacting with these replays, whether it's a non-spatial, some other orthogonal representation of experience, it could allow for this representation to have either more time for computation or have a more textured, nuanced representation of experience. So that could be a potential reason to want to have these longer replays. So early on in learning, you have a gist that just gets you across the track, and then later you have this detail. You've added these new hover locations. You've added some detail and maybe a cortical representation that's more detailed along with it. So that's all I'll show you today. So I showed you that replay appears after a single experience and lasts, irrespective of where the rat is. That significant replay of a local environmentally novel space will slow down with experience, and that these replays slow down by adding detail into them, by adding these attractors perhaps. And then I showed you a potential mechanism of increasing inhibition and a potential consequence of cortical engagement for this effect. So yeah, I wanted to bring up that this could have implications for memory. This is a recently published paper from the Bajaki lab showing that when you artificially extend ripples, which are associated with replay, you actually get a benefit in memory here. These are outbound trials on a delayed alternation task. So maybe another reason to have these longer replays, this cortical engagement or whatever else is, or just having these more detailed representations might actually be aiding the animal during that environment, during that task, aiding their memory online. And with that, I'll just want to thank all the people who collected all this data and helped me with this. So Ting Feng, a former graduate student in the lab, did a lot of early data organization and had a lot of early ideas, got the ball rolling. And then Delia Silva did the novelty recordings, a whole bunch of really nice novelty recordings. Jiajing Wu did this PFC hippocampal recordings. Alan Ambrose did the increased reward experiment, and Brad Pfeiffer did the single app experiment and some of the reward experiments too. And also, I'd thank the lab that currently 3 postdocs and 2 grad students, and thank you all for your attention. And do questions.
Great, thanks very much. That was really interesting. So yeah, I think people actually asked. We ended up asking questions throughout, but yeah, here there's one question. Actually, 2 questions. So, do reverse replays also slow down? That's one question. And the second question: have you looked at sleep and what happened in sleep? Does slowing happen during or following sleep?
Great questions. Okay, so yeah, I broke down this effect into forward and reverse, and reverse replays definitely slow down. But so do forward replays. There's more reverse replays in my data set, and there's no differences between reverse and forward in terms of how much they're slowing down or the rate at which they're slowing down or anything. So it looks like both forward and reverse replays are slowing down, and that there are a lot of reverse replays in this data set, so I can definitely confidently say that reverse replays are slowing down. And then the sleep question, definitely, I'll just go back here. So yeah, I looked during sleep, either after a single experience or after an entire session. And in both cases, I did not see that the replays were slowing down. The duration wasn't changing and the slope wasn't changing. So I don't think that replays are slowing down across sleep. One kind of related question also could be, what happens on the second day? With even more experience, on day 2 after a night's sleep, do they continue to slow down? And I have kind of looked to see whether maybe there might be a U shape going on, like maybe, this effect starts turning around or something. I don't have enough data to speak to that. There's just not enough sessions on the same track on subsequent days that I can really look at a curve that goes out that long, but it's something that I think is could be interesting. And there are other findings that kind of make it seem like maybe it might be that they don't continue to slow down. So.
Cool. There are a few more questions. I'll ask the ones that are in the chat first, and then we'll go to inviting people. So one question is, the control of remote replay is really nice. When you compare those from the perspective of the LFP, are they different in terms of gamma, the sharp wave, and the physical duration?
So are remote replays different in any LFP signature or in the duration? I don't think that they are different in the duration, like as a main effect. I don't think I saw that. I've not looked at things like whether the gamma, I've not looked explicitly at whether things like gamma or the sharp wave or things are different, whether you could, there has been work that sort of, I think maybe if you had enough data, maybe if you had probe electrodes, you had enough data across the hippocampus, maybe you'd be able to pull out some things that would distinguish ripples of remote versus current environment. But I've never seen anything like that in my data. But it's a cool idea to look at.
Cool. Next question is from Anna. Do you also see incorporation of additional attractors into the non-significant candidate spike density events?
So I don't see that those events slow down. So they don't increase the number of steps. So they don't increase the number of hovers. But I have done the analysis that I showed you 2 different ways when looking at the distribution of hovers, because the way that I think about the attractors, the way that it would work within the system, I think that all kind of activity would, if it's moving around in this space, should be, all activity would be falling into these attractors, even if it isn't classified as a significant replay. So I looked at those hover distributions both with only significant replays and with just all of the candidate events. So not taking just the not significant ones, but just dumping all of them in, significant or non-significant. And the results are the same either way. So you still have all of those effects that I showed you before about the fact that they don't change across experience, and that you're adding a hover location, you're inserting it in the second, that you need a full lap to get more attractors. If you include any type of noisy spike density events, you still don't have those inserted hovers, basically, is another way of saying that. So I haven't looked at comparing hovers in the noisy ones versus in replays, but it looks like they're both behaving within the same system of attractors, in that they need to have experience in order to dwell in a certain place.
Thanks. I think the next question is, sorry if I'm missing some, I'll find them. So one question is, do you think the same thing will happen in theta sequence?
Yeah, that's a good question. And it's like something that's on my list of things to look at that I haven't gotten to. I don't think it would be as simple as that it would happen during theta sequences. I mean, yeah, you have gamma during theta, and you can have these sort of nested sequences within gamma. I don't know. It's an open question. I don't know. It's a good question.
Do slow wave sleep replays slow themselves? So not whether the explanation of the question, not whether sleep changes wake replay, but whether sleep replay is changed.
Right. So no, it doesn't in this data. So after just one pass across the lap or across or after a full session, the replays that occur during the sleep after an experience don't get longer on their own. Yeah, needs experience for them to slow down.
Did you see any correlation between higher place cell stability and increase of replay during across laps? Maybe using tags information or a similar measure?
So I haven't looked at that exactly, but what I will say, I did look at, I at one point had the idea that maybe, you know, during this novel experience, they're going to have, you know, place cells coming online and also place cells moving around. So I thought, well, maybe there could be that the place cells that are active on the first lap and then that are active during these first rapid replays, what if those are the cells that are like a backbone of all future replays, and they like participate in all replays, and these other cells that are coming online are like filling in the details. And I looked to see whether that was true, and I did not see any evidence of that. What it looks like overall is that the replays that tend to fire, the cells that tend to fire in replays tend to be the cells that were recently active. So no matter whether it's the first lap or the 14th lap, if you look at how cells participate in replay, they're more likely to participate in the replay if they were more active in the last two lap or so along the track. So it looks like all the cells are participating similarly within the replays. It's just the network of the replays that are changing. It kind of doesn't answer your question, but it kind of touches at it a bit. You can requalify and ask me another one if you want.
Cool. Is it really an increase in inhibition with experienced, i.e., if you look at change in firing rate in pyramidal cells, do you see the same increase as you see for the interneurons? Or if firing rate doesn't change in pyramidal cells, more pyramidal cells firing may be enough to get increase in firing rate in the interneurons?
Yeah, I don't see changes in firing rate as a whole during this in pyramidal neurons. So I think, so I don't think that it's just that the whole network is. I think that the pyramidal cells are remaining constant, but it's kind of, I don't know. It is a bit. I have some kind of questions about that. It is kind of tricky to look at, but yeah, it looks like the pyramidal cells are behaving similarly, like same, same firing rate within replay, even though the cells, they're firing more on the track across time. Within replays, they seem to be constant firing rates.
Thanks. Can you look at these attractors during theta or exploration to find out whether attractors form online or offline? Okay. And then Muhammadi noticed that Yunza actually asked the question about theta before, which I noticed while I was reading it. And great. So that was answered. Is this CA1 only or CA3 too? And what do you see or expect to see in CA3?
Yeah, this is CA1 only. This is all dorsal CA1 recordings. And yeah, it would be really cool to look at this in CA3 and see whether what the attractors are like in CA3. I would expect that they are also slowing down, but I I don't know. Yeah, we don't have any of that data that I could look at. We do have data where CA3 was inhibited, and there's somebody in the lab looking at that data set, looking at like about details of gamma within these things, but I don't think that they've looked at hovers with a change to CA3. So it would be really cool to look at, but I haven't had the data to look at it.
Great. I think that's all the questions. Yeah, so thanks very much. It was an excellent talk. And now everybody can go and swim in the river or something. Yeah, so thanks for all coming around, and there's a round of applause in the chat. So
thanks. Thank you everyone for your attention. This is fun. This is great. Yeah. Thanks for inviting me.
Cool. All right, I'm going to sign off now. The chat is still active if people want to.
Cool.
Yeah.
That's very cool. I'm going to read it a bit. All right.
Have a good weekend.
You too. Bye-bye.